guys, welcome to Biopsy. Today we are going to discuss about tumor suppressor genes in RB gene and TP53 gene. Okay, but let's revise first about hallmarks of cancer. There are total 8 hallmarks. First is the self-sufficiency and growth signals. Second is insensitivity to growth inhibitory signals, which we are going to study today. And third is altered cellular metabolism. Fourth is evasion of apoptosis. Fifth is limitless replicative potential. Sixth is sustained angiogenesis. Seven is ability to invade and metastasize. And eight is ability to invade the host immune response. And let us also just talk about a little bit cell cycle and regulation. As we all know, there is checkpoint 1 between G1 and S phase, and which is regulated by cyclin D and CDK4 and CDK6, which promotes the cell cycle to continue forward. Okay guys, so guys, let us talk about tumor suppressor. So what are the tumor suppressors? They are simply a protein or gene that opposes any of the various hallmarks of cancer. Yes guys, these are the genes or proteins which opposes the various hallmarks of cancer. So there is RB gene and RB and P53. They stops and shut downs the cell cycle, while other tumor suppressor genes, which we are going to study further in next video, they are also transcription factors there. So cell cycle inhibitors are there, signal transduction molecules are there, cell surface receptors, some of them are cell surface receptors, and regulators of cell response to the DNA damages. But we are going to study them in the next video. Okay guys, let's start our talk first with the retinoblastoma, or governor of the proliferation, RB. Okay, this name is important. Many MCQs are there on this name, which is governor of proliferation, which is retinoblastoma. So first, let's as we all just saw that the G1 and S phase, there is between checkpoint 1, and there is cyclin D and CDK4 and CDK6, which promotes the cell cycle to continue. Well, this retinoblastoma gene, what happens? Normally it is in hypophosphorylated state, guys. Yes guys. So in hypophosphorylated state, it is active RB, and RB protein inhibits the checkpoint 1, inhibits the cell cycle there and stops the cycle there. Yes guys, what does this hypophosphorylated RB protein does? It binds to the E2F transcription factor, and as it is binding to the transcription factors, so transcription obviously would not be able to complete or move forward, so the cell cycle would be inhibited there only. Now guys, what happens? This cyclin D and CDK4 and CDK6, when they are increased, they want cell cycle to be continued. So what does this cyclin D and CDK4 and 6 do? They causes the hyperphosphorylation of the RB protein, and because of hyperphosphorylation of the RB protein, this RB protein gets inactivated, so it cannot inhibit the cell cycle more. So cell cycle continues and cell enters in the S phase. And there are CDK inhibitors, P16 and INK4A. These inhibit cyclin D and CDK4 and CDK6. So what does this CDK inhibitors do? As they inhibit these guys, so hyperphosphorylation of the RB is not possible. So RB would be in hypophosphorylated state, and it will be in active state. So it would be inhibiting the cell cycle. Yes guys. Now, if due to loss of function of RB mutation, what occurs here? Obviously there will be tumor occurring. Which tumor? Retinoblastoma. It is the most common tumor, and osteosarcoma can also be occur. Now guys, this retinoblastoma tumor, it is transmitted as autosomal dominant trait. Now as we will discuss this right now, that how does it transmitted as the autosomal dominant trait. Okay, there is two-hit hypothesis in RB. What happens in normal individual? Let's assume that this first figure, it is of a normal individual. There is normal female, this is normal male, and they fertilize the cell, and many cells are there, and ultimately what happens, due to some environment factors or any other factors, the mutation in one gene occurs. This is first hit, guys. This is first hit. Now as you all know, this one gene is mutated, but the other gene is right now there, which is working perfectly. isn't it guys? But now after some time, or after due to some environmental factors or some under radiation or whatever is there, due to that this second gene, it also gets mutated, and as this both the gene gets mutated, there is loss of function of RB. Yes guys, RB gene. So there will be retinoblastoma formation. This is in sporadic form. This is sporadic. As you all know, there is second hit here. Second hit. Now to get two hit in the same gene, it is a little bit it is a hard thing. It is not common that much. So it is rare. It is not that much common. Well, what happens, like this parent has already one mutant RB gene there, and it is transmitted to its child. Okay, and this mutant RB in cells, there is one gene mutated and other is functional. So this genome needs only first single hit. As there will be single hit, the other gene also would be mutated, and it will cause the retinoblastoma or the tumor cancer. So and this getting one single hit, it is very easy, or you can say it is very common to get one hit, but it is not very common or rare or uncommon to get the double hit on both, to get the both the genes mutated sporadically. So guys, this is transmitted as autosomal dominant trait. Now guys, at the level of individual cells, as you guys can see here, individual cells, RB loss of function behaves like a recessive trait. Yes guys, that could be understood. At the level of cells, it is acts as recessive trait, but at clinically, or as we can say in environmentally or our surrounding, it acts as autosomal dominant, but at individual cells it is recessive trait. Now guys, there is other thing that there is a line stating that transforming proteins of several oncogenic animal and human DNA viruses also neutralizes the growth inhibitory action of RB or retinoblastoma. Now how can we understand this guys? Like there is HPV16 virus, and this HPV16 virus has a E7 protein component. This E7 protein component that binds with the hypophosphorylated guys. Hypophosphorylated means active. Yes guys, binds to this active RB, and it releases the E2F transcription factor. Obviously RB is binding and inhibiting the E2F factors. As E7 is bound with the hypophosphorylated state, now E2F transcription factor would be free, and as this would be free, it would causes cell cycle progression or cell cycle from G1 to the S phase. It will from G1 to the S phase, cell cycle will go and cell cycle would progress. Yes guys. Now finally let's summarize. Anti-proliferative effect of RB is lost in cancers through this mechanism, which these four main mechanism. One is the loss of function RB retinoblastoma mutation, another is amplification of CDK4 and cyclin D genes. Yes guys, CDK4 and cyclin D genes will be amplified, then retinoblastoma would not be enough to inhibit them. Then there will be loss of function of mutation affecting cyclin dependent kinase inhibitors P16 and INK4A guys, as we have seen here is P16 and INK4A. These are CDK inhibitors. They inhibit the cyclin D and CDK4 and 6. Yes guys, so this is done. And the other fourth is viral oncoproteins that bind and inhibit RB E7 proteins of HPV, as we have talked here. Yes guys. So guys, let's talk about the other tumor suppressor, which is TP53, and it is also known as guardian of genome. Guys, these names are important. This TP53 is known as guardian of genome, while retinoblastoma was known as governor of proliferation. Okay guys, so let's talk about the TP53 gene. So this TP53 gene, it is located on the chromosome 17p13.1. Yes guys, what does this TP53 do? As it is known as guardian of genome, what does this TP53 gene do? It regulates, it doesn't help in, it regulates the cell cycle progression, DNA repair, cellular senescence and apoptosis. Yes guys, there is one terminology in TP53 gene, Li-Fraumeni syndrome. What is there in Li-Fraumeni syndrome? It is nothing guys, it is just that individual have one inherited mutated copy of TP53 gene. Yes guys, there would be one mutated copy of TP53 gene which would be inherited in any individual, due to which what happens, there would be increased chances of getting cancer. And which cancer guys? There is sarcomas, breast cancers, leukemias, brain tumors, and carcinoma of adrenal cortex. So in Lee-Fraumeni syndrome, these cancers are common. So guys, let's talk about TP53 gene. What does this TP53 gene do? As it is a gene, transcription would be always obviously there, so it will be coding something. What does it code? It codes P53 protein. And what does this P53 protein does? It will do something. We will study the work of the P53 protein, but in normal circumstances, this P53 protein is formed and it is bound to MDM2 proteins, and this MDM2 proteins causes degradation of P53 proteins. Now guys, if there is we can think that if there is already mutation in TP53 gene, there will be loss of function obviously. What other thing can happen? Overexpression of MDM2 protein happens, then it would obviously clear off all the P53 proteins, or sometimes mutation of genes encoding this protein also can happen, which will cause increase in P53 or accumulation of P53 in the cells. Okay guys, so let's talk more. In normally P53 is bound to MDM2 receptors, so it is undetectable. We cannot detect in normal cell P53. But what happens in stress? As there is stress to any cell, it will be released from MDM2 receptors. And how it will be released? There are two mechanisms from releasing it from MDM2 receptors. One, let's talk about the first one. What occurs? DNA damage and hypoxia. Due to DNA damage and hypoxia, there is increased in ataxia telangiectasia mutated and ataxia telangiectasia RAD3 related. These protein kinases are increased, which causes the phosphorylation of P53 and MDM2. This phosphorylation, and there is phosphorylation of MDM2, the degradation would be obviously inhibited or it would be stopped. So P53 would accumulate in cell. The other mechanism is that when there is oncogenic stress in the cell, as there is oncogenic stress, how does oncogenic stress? Activation of such oncoproteins such as RAS. So due to activation of RAS, there would be cellular stress, and this as there would be cellular stress, it would activate CDKN2A, which is a tumor suppressor gene, and this CDKN2A will encode code for P14 and ARF, and this tumor suppressor this P14 and ARF, they bind to MDM2 receptors and displaces P53, which causes accumulation of P53 into the cell. Now guys, as P53 is accumulating in the cell, what does this P53 do? That is a big question. So let's know what does this P53 protein do. First thing, P53 mediated cell cycle arrest. When there is early rise of P53, or there is low concentration of P53 is there in the cell, it will be obviously primary response to a DNA damage. There will be stress, so P53 will be increased. So as P53 is slightly increased, it will causes CDKN1A gene transcription, and this CDKN1A gene will code for P21 protein. This P21 protein inhibits CDK4 and a D-cyclin, and due to which transient cell cycle arrest occurs. And as there is cell cycle arrest, the cell will get some time. It will get some time and it will repair its DNA. And as the DNA is repaired, this P53 level would fall and cell cycle would be continued. The other thing is that as this P53 is activating CDKN1A gene, it also induces GADD4S protein, 45 protein. So what does GADD mean for? Growth, arrest and DNA damage, and this protein will causes DNA repair. Yes guys. The other thing, what if this DNA is we can't repair this DNA, what would happen? So P53 will more increase, it will be increased intermediate rise in the P53 concentration in the cell. So this P53 will causes epigenetic changes, and due to which permanent cell cycle arrest will be there. So the cell would not be able to proliferate anymore. And and at last, when P53 is accumulated more, like there is very high concentration of P53 in the cell, it will activate the pro-apoptotic genes like BAX and PUMA, and these BAX and PUMA will cause apoptosis. Okay guys, let's understand that the same thing with this diagram. As there is ionizing radiation carcinogen mutagens, it would cause stress in the cell. Is one cell is this cell is normal cell, while this is mutation or loss of P53. Yes guys, and normal cells, as their DNA damage occurs due to which oncogenic stress or hypoxia, and P53 accumulates and binds to the DNA. This P53 transcription dependent and independent effects on targets. One, what we would call senescence, so cell would be arrested their cell cycle, it would not be able to proliferate more. Others, it would cause transcription of P21 protein, which is a CDK inhibitor. It will cause G1 arrest and causes their and helps in repair of DNA. Other, GADD45, which causes DNA repair, and successful repair, the cell cycle would the cell would become normal. And guys, if the repair is not possible in the DNA, then it would causes pro-apoptotic gene activation, BAX and PUMA, and it would cause apoptosis of the gene. Now guys, let's see what happens if there is mutation of loss of P53. Yes guys, so there will be DNA damage, but there is no P53 dependent gene activation, so no cell cycle arrest would be there, and no DNA repair, and no senescence would be there. So these mutant cells will continue growing, continue replicating, and it will cause a form of malignant tumor. Yes guys, that is how P53 is important in regulating our cell cycle and preventing from neoplasm. Okay, as we talked in the retinoblastoma, there was E7 protein of the HPV, which was binding to it and inhibiting it. Okay, yes guys, so this P53 protein, there is E6 protein of high risk HPVs, and which binds to P53 and promotes its degradation. Guys, this HPV stands for human papilloma virus. Sorry, I forgot to mention it earlier. HPV is human papilloma virus, and it will bind to P53 and promote its degradation. So as P53 would not be there, so there would be no arrest in cell cycle, and it will cause neoplastic growth of any cell. Yes guys, so other tumor suppressor genes we are going to study in the next video. Thank you for watching guys. If you liked our video, please like, share and subscribe. You can check in description for our notes, and thank you so much guys. Have a good day.